functions are very heterogeneously progressive. As you can imagine, once you start to have a white hair, it's not coming from all along. It's the one this side and the other one, the other side is very heterogeneous. That's exactly what's happening to RP cell within the retina throughout the stress and aging mechanism. So our group at the USC and then a few other groups in the globally did RP cell transplantation trial. So due to the technological development in stem cell world, now we can differentiate the RP cell, healthy RP cell, from either human embryonic stem cell or pluripotent stem cell. And our group, over the decades, we focused on human embryonic stem cell derived RP cell, and then we made this patch technology, and then we just completed a phase 1/2A trial several years ago on 16 patients. That outcome was pretty impressive. Not only was it safe within the eye, although it was not immunologically matched, but also we observed several signals in terms of improving vision, even if the photoreceptors RP cells are gone in this central geographic atrophy area. And several hypotheses that we had was not only the cells implanted were working, but also we thought that the cell-secreted secretome was doing important work in terms of paracrine and autocrine action. So this study is going to launch phase 2B trial where I'm going to decide PI in USC, in addition to several other studies in the United States later this year. But as an EV scientist, I was also wondering, how about using these cells-secreted EV and what they could do? So first of all, I wanted to, isolate the EV from this RP cell. We used, as I mentioned, human embryonic stem cell derived RP because we already have a safety profile within the eye in human trial. And I also wanted to figure out how can I isolate the EV that is applicable for clinical trial moving forward. So we all know differential ultrasonification, there's a limitation for that. So we had used ExoDisc, which is kind of modified tangential flow filtration system. I have no financial interest on this, but I thought that this could be potentially modeled to test the TFF system moving forward in clinical trials. And I wanted to compare with the differential ultrasonification. And I also realized that over the time, not any of the device was all the times right. So I also wanted to compare multiple devices to characterize our EV. That was really the message that I have gotten from the MICEP 2023 publication as well. So we measured our particle and size using 3 different machines, NanoSight, CW, flow analyzer. I'm sure everybody has some experience in one of this, or at least all of this. And also we tested surface epitope. Not only we just did a Western blot in terms of yes or no, we also characterized them in a very comprehensive way, which I'm going to introduce a little bit. And then we use the TAM for morphology analysis. First of all, we also characterize the cell clearly so that we're dealing with the RP cell, because once you're going through the differentiation, the cell could be very heterogeneous in terms of your culture place. So you want to make sure that your cells are fully differentiated and they have full markers for this RP cell. So we isolated EV from 2 different machines, and then we measured them with 3 different devices. As you can see, the particle numbers and size are actually variable. Not only variable between those devices, depending on how we recover the EV from ExoDisc versus ultrasonification, these 3 machines are also showing us different particle numbers and size, and then the coefficient variance was also variable. I'm not, conveying one machine is better than the other here, because I really believe that depending on what type of EV, every investigator needs to re-verify their workflow, which one is better than the other, because at least we confirm that there is some consistency between the devices as well. So you have to just understand whenever someone say 20 million. Is it really 20 million? What machine was measuring this 20 million? And some investigators says, we did a dosing test of 1 million versus 3 million. But 1 million versus 3 million could be easily coming from same devices or different devices. And we also tested scalability depending on two different strategies. 10 ml input versus 40 ml input. Are they really proportionally increasing in terms of particle numbers? And then, as I mentioned, we comprehensively characterize our EV. So as you can see, I'm going to just show this barcode that we did with multiplex, maxplex, I believe, with the first one. The patterns are similar between ExoDisc and then ultracentrifugation, which is comparable to Hexcel. I just want to make sure that not all EVs are having the same barcode. So one of several undergraduate students in my lab, they know, oh, this is RP cell EV signature versus this is other cell type of signature because it's persistent. So my lab is moving forward using this strategy as the index characterization. Now, how we're going to deliver this EV? So intravitreal injection is very common, the most common actually procedure done globally in anywhere in your body. And in the United States, only 7 million injection per year is done just to deliver various types of medications. So I believe this is one strategy to begin with, although we have to figure out how to deliver this in a sustainable way. But one of the challenges that we have to validate is not all the medications are penetrating the entire retinal thickness. This retinal layer has multiple layers of cell, and then also there is a blood retinal barrier does exist. So several years ago, we reported that intravitreally injected EV can penetrate the full retinal thickness, and also they can bypass blood retinal barrier, which is similar to that has reported that EV can bypass blood brain barrier. Back then, we actually used the PKH stain, but it turns out to be a PKH stain has some limitation. Now we're using Man-Blue, I believe, and then we tested the same intravitreal injection and then full retinal thickness penetration using the Man-Blue, and then using this particular EV, and we confirmed that they can penetrate the full thickness retina. Now, how are we going to demonstrate that this RP cell-derived EV could be therapeutically useful? First of all, you have to find also the right model to demonstrate. So RCS-RED actually has a multi-K mutation in RP, therefore secondarily photoreceptor side. What the multi-K does is, as I mentioned, photoreceptor outer segment has to be eaten by RP cell every 10 days. But due to the multi-K mutation, photoreceptor outer segment cannot be digested by RP cell. Therefore, both cells are actually degenerated. And then this is a perfect model to test the functionality that I'm proposing of RP cell-derived EV. And then we injected this EV to RCS-RED. We demonstrated that photoreceptor was rescued, and then also the photoreceptor functionality, which is measured by EIG, was improved after the treatment. So how did this happen? Because I just provided RP cell-derived EV to RP cell mutation model. There are multiple ways to look into this, but I want to have a picture here first. We did work RNA sequence transcriptomics, and then obviously whenever you compare treated versus non-treated, you can always have some differentially expressed gene. But my interest was, which direction are we going after treatment? Are we going back to the normal level? So we compared this transcriptomics compared to normal retina. So as you can see, compared to normal retina, RCS-RED retina has a lot of genes that downregulated and some upregulated. That entire changes were actually reversed toward the healthy retina. So actually, statistically, 88% of the gene that are expressed in the retina going back to toward the normal retina. One of the reviewer actually says your changes are very minimal, but this is just not all optimized treatment. So, but the surprising fact here is that the EV treatment is not touching several pathways. It's actually touching general pathway within the retina. So that's the key message here. And then we look into multiple pathways, which pathways are more important in this therapeutic mechanism. As I mentioned, the photoreceptor outer segment phagocytosis was detected in this model. However, we confirmed that several other genes that are involved in photoreceptor outer segment phagocytosis are upregulated, although just RP cell SAB was treated. And then that was a significant. And then also many genes, as you can see here in the red box, related to photoreceptor transduction and a vitamin A cycle, which is the visual cycle that are critical in photoreceptor light processing, are upregulated by this treatment. And also, we also found that the lipid metabolism, this is a little bit more complicated story, but at least you can just think that the lipid metabolism is one of the important key function of the RPE, and then RP-derived SAB treatment helped this lipid metabolism genes in multiple levels. What about the reactive gliosis? So as you can imagine, all this retinal degeneration very similar to Alzheimer's type of disease. Each time the cells are dying, reactive gliosis, upregulated. And same thing with the RCS red, and the RP cell secreted EV treatment actually reduced reactive gliosis. It's like a global therapeutic benefit, as you can see. And the other thing that I really wanted to answer was we treated human cell-derived EV in mouse or rat. They immunologically safe because when we did a human trial for stem cell implantation trial, the patient had to take the immunosuppressants for the time being because they're not immunologically matched. But in this case, at least during one injection, I believe that this is one injection, and then we checked the 1 day and then 7 day after, and then we did not see any significant immune reaction. However, we think that we should check the long-term therapeutic consequences if there's any immune reaction. That's next study that we're working on. And then now everybody was asking what is in RP cell-derived EV. So we first checked the microRNA because a proteomic study has been done in the past, just looking at overall RP secretome. microRNA is relatively new, and I think the microRNA is really relevant to more EV. Therefore, we profiled EV. Actually, we compared EV profiling between different passages of the cell because not all passage cell at different conditions are containing the same EV, which is my hypothesis. Therefore, we checked 3 different passages of the same cell, and we confirmed that their microRNA profiles are different. And also, what is those microRNA's function? We confirmed that those are EV trafficking, tissue and cellular polarity, which is RP cell itself, the maintenance. In addition to growth factor, inflammation, oxidative stress, and synthesis are more RP functions. So therefore, RP secreted EV has not only the resin microRNA that is necessary for EV trafficking and their own cell maintenance, which is tissue cellular polarity. It actually also contains the microRNA that are necessary for growth factor, inflammation, oxidative stress, and synthesis, which reflect the RP function. So what's next? So we have some idea how this works and what molecules contain EV did this, but it's going to take a much more complex, comprehensive study through the multi-omic study. One of the challenges that I'm encountering, I'm hoping that I can answer, I can hear answer from some investigator is this is going to be multitudinal interaction. So molecule A and B come together to pathway C. So it's not just the X to Y axis. We're talking about 10 different axes working together. And I don't know who can analyze this for me. I think perhaps AI can do this, but this is really that something that we need very eminently from the bioinformatics side. So tachyonosis here, EV has a potential to restore cell functionality. We've been so focusing on regenerative or anti-inflammatory, but actually once you pick the right cell as an EV source, they can potentially restore to cell functionality. And TFF is a valuable strategy for separating EV for the development of intraocular therapeutics. And using all the manual approach in EV characterization remains ideal for reliably characterizing heterogeneous EV hopefully we can have a better strategy to analyze in a single cell level for the time being. This is how we manage this. And then lastly, multi-omic study may be helpful to correlate the action mechanism and active molecule from the EV treatment. I'd like to thank the Regulatory Affairs and Clinical Use of EV-based Therapeutics Task Force led by Eva, and then multiple members are really brainstorming how to move this field forward. I really want to see many EV therapeutics are treating patients in the near future. And then these are my collaborator, and then these are my fundings, and these are my lab member. Thank you.
Okay, so thank you, Dr. Sun-Li Yang. It might be a good news for you that due to a fault of myself, we have one talk less. So we will be 20 minutes earlier ready, and I can release you in the evening. But now we have 5 minutes for question and answer. I would say we make it immediately as we have a bit more space. And if there is a burning question to Sun-Li Yang, who is really doing a great job of harmonizing and combining those scientific concepts with the opportunity to use them in the clinic. If there's anything which popped up. Takyan, please.
Thank you for a really wonderful talk. So it looks like the effect seems to be just a cell survival issue, right? When
I think so the RP cell you can imagine as a factory. So at the end of the disease, RP cell die,
right.
And I don't know how much RP secreted EV could help in this situation. But during that time, as I mentioned, RP dysfunction is very long, heterogeneous process. They're struggling for many years during the disease process. So if you provide the RP secreted EV, it's like you're trapped in the mountain, but somebody gives you food and water as a maintenance, so you can survive longer.
Right. So in some ways, like aging, right? It's a long,
It's a long
process.
Yes. Mm-hmm.
Now, so MSC EVs do seem to have this anti-aging effect. So my question is, would that be possible to use MSC EVs rather than just RP EVs?
Yeah, there are actually several investigators who investigated MSC-derived EV, but more targeting for uveitis, which is inflammatory disease. What I envision this is, yes, right before RP cells are fully differentiated, some of the early stage semi-pluripotent cell or predetermined to become retinal cells are actually currently under clinical trial, putting in the eye, see if they can be regenerated. There are some issues we have learned from the stem cell treatment several years ago. A lot of investigators just inject whatever stages of the cell in the eye, and then they become really tumorigenic, and then scar formation. That was a huge, actually, news platform in New York Times several years ago, and the patient became blind, actually. So within the eye, it's pretty delicate that not only functionality, but also, let's say you rescue the cell, but then somehow that treatment induced the scar formation, essentially destroyed anatomy, and then the functionality is no longer valid. So you have to rescue the cell without pretty much no inflammation, which is the key here. And MSC cell does not exist in a physiologic state within the eye. That's my argument. But my best friend is studying MSC EV in the eye to treat the uveitis. So this is a great question, and I think everybody should study whichever they want to, but based upon very strong scientific premises, not because the other investigator uses this cell, so I'm going to use this cell, or accessibility or proximity. I think everybody should have a really clear scientific premise.
Thank you.
Thank you. It was a wonderful talk. I actually have a friend that's taking MSC exosomes, and her uveitis remitted. But that's just an N of one. But the question I have for you is, well, I have a couple of questions. So dogma that the retinal pigment epithelium is finite. Is that true? Because they say that about the neurological, system, but that's clearly not true. Could there be some stem-like characteristics that replenish the RPE? I mean, because tonically there's inhibitors of dedifferentiation that can be blocked with microRNA throughout the nervous system. So,
yeah, those are all we will need to study. I don't think microRNA is the only active molecules that are working here. But I think microRNA gives us an idea how this multiple functionality can be restored. And when we did the functionality analysis, it really reflects the cell, the mother cell who's secreting AD, because different passage and different profile. And for example, I didn't explain too much because of the time strain. So for example, only passage RP cell didn't have much functionality related to microRNA. It's more about actually EV trafficking and RP maintenance. The intermediate stage has a lot more enriched microRNA profile. And later stage also reduced their profile compared to what the earlier stage. So I think there is really tight regulation in terms of their cell conditions and cell stage, how they secrete the microRNA in the EV, which suggests that depending on what stage you're looking at, condition you're looking at, their functionality could be different.
So, thanks a lot, San Liang. And now it's my pleasure to announce Dr. Naomi Shiraoki from Waseda University in Tokyo. She brought her own laptop and while she's installing, we are looking forward to your talk where you are investigating the impact of surface and environmental factors on cancer EV delivery.
Thank you very much for your introduction. And good evening, everyone. I'm Naomi Shiraoki from Waseda University, Tokyo, Japan. And it's a great honor to present my work here. And I'd like to thank both organizers for this opportunity. And because I'm a cancer researcher, so today I'd like to talk about how EVs are transferred within the tissues and incorporated to the cancer to the target cells from the cancer research. So the solid tumor tissue is not most of the cancer cells, but also consists of a variety of non-cancerous cells, such as fibroblasts or immune cells, or endothelial cells, and also non-cellular components like an extracellular matrix as a scaffold. And these cancer cells interact with the surrounding cells through these direct interactions or like secrete factors, including extracellular vesicles or EVs, to modulate them into the tumor support phenotypes to establish the tumor microenvironment which support the cancer growth and metastasis. And further, the secreted cancer EVs travel systemically via blood or lymphatic flow to reach to the distal organs and alter the distant organs to environment to establish a premetastatic niche and support the cancer metastasis. So in my previous study, I looked at cancer EVs using the antibodies in this mouse breast cancer model and to show the suppressing the lung metastasis and proving the therapeutic concept of the EV targeting therapy. And because EVs transmit intercellular signals by direct interactions or being incorporated to the recipient cells, so it's really important for EVs to reach to the right target cells to function properly. So indeed, that several studies, including today's sessions, show that cancer EVs have preferences in the recipient cells, and also the cancer EVs has organ tropisms to prime the cancer metastatic niche at distal organs. But the exact molecular mechanisms of these direct targeted delivery of the EVs are only partially understood. And previously, my former colleague, Dr. Tominaga and Dr. Ochia's group established the brain metastatic human breast cancer cell line BMD2A and B through the repeated seeding and selection within the mouse model. And the selected BMD2A and B enriched the microRNA 181C in their EVs and break down the BBB to promote the brain metastasis. And in this study, the BMD2A and B EVs are more incorporated into these endothelial cells in this cultured BBB model compared to these parental cells. And when they are treated systemically, the more EVs are accumulated to the brain compared to these parental cell EVs. Later on, I analyzed the glycosylation patterns of these EVs from the BMD2A cells. And altered glycosylation patterns are a very common event in cancer, regulating the cancer progression and metastasis. And many reports have shown that the surface of the EVs are highly glycosylated. And the glycosyl chains on the EVs are attached to either proteins via N-linked or O-linked glycosylations towards the amino acid side chains, or lipid like glycolipids, including sphingolipids. And I ran the lectin blot analysis to analyze the glycosylation patterns of the BMD2A EVs and found that they have the enriched N-linked glycosylations recognized by these two lectins, whereas they decrease the O-linked glycosylations, including sialic acids, recognize these lectins. And then we analyze the functions of these surface glycosylations of the cellular uptake. And first, BMD2A EVs or other parental EVs are stained with PK67 and removes both N or O-linked glycosylations intensively using the enzyme and supplemented to the human endothelial cells. And you can see that removing the surface glycosylation actually stimulated cellular uptake, like QX. And also I tried removing either N or O-linked glycosylation using a specific enzymes. And you can see that removing either of the glycosylations are enough to enhance the cellular uptake of the EVs. Then we analyzed the roles of the surface EV glycosylations on biodistributions. And EVs are removed with BMD2A EVs are removed with O or N or O-linked glycosylations or both. And you can see that removing the O-linked glycosylation stimulated the lung accumulation of the EVs. And also for brains, you can, but not in N-linked glycosylations. And for brains, you can see that the O-linked glycosylation removal slightly increased the accumulation to the brain, but it was not statistically significant. So together with the in vitro data previously, these results shows that the surface glycosylation of the EVs have suppressive roles on the cellular uptake, and especially O-linked glycosylation have the impact on the biodistribution of the EVs. And we also assume that BMD2A EVs reduce the bulk glycosylation of the surface, which helps the EVs to reach the brain, reach and accumulate brain to stimulate the brain metastasis. And for next step, I think that we have to analyze the specific glycosylation patterns for specific roles of the specific glycosylation structure on the cellular uptake and other biological roles. And for that, we need a specific enzyme that trains the specific structure of the glycosylation. For one of them is EGCase, which can remove the glycosylation from the glycosphingolipids specifically, and this kind of specific enzymes will become a useful tool to investigate the roles of the biological roles of the functions of the specific structure of the glycosylations. Then here I will switch a gear a little bit to discuss how the cancer EVs are transferred within the tumor tissues. And this is a microscopy image of the colon cancer patient tumor from the Dr. Yon Lo's group's paper. And you can see that red arrows are all EVs, and you can see that the EVs are secreted to this narrow interstitial space. And as this schematic shows, that the tumor tissue have a very highly packed structure with a very unique tumor microenvironment. And cancer EVs secreted from these cancer cells are surrounded by this variety of the surrounding cells, non-cancerous cells, and also with the ECM. So therefore, in that situation, the tumor tissue environment will affect the EV transfer within the tumor tissues. And I think that I assume that there will be several impacts on that. And one would be like altering the EV properties, like changing the amount or size or cargos or their surface properties, or also the target cells, because not all the cells within the tumors will uptake the cancer-derived EVs at the same level. And also the distribution or like the distance of reach, because all the tissue structure will pose the physical barriers to the EV transfer. So to truly understand the cancer EV transfer within the real tumor tissues, we need tissue-based experimental systems. And here I decided to use this tissue slice culture system to study the cancer EV transfer within the intact tumor tissues. And tissue slice culture is a thinly sliced tissue that maintains life in the lab with the culture medium and also the support of the cell culture insert. And this tissue slice culture maintains their original cell tissue architecture and also the cell profiles, and can be used for like tissue models for many kinds of experiments. And this tissue slice culture can be prepared from like many like solid tissues, including tumor tissues and like organs, mouse models, or even from the patient tumors. Here's an image, a movie of this tissue slice preparation. And using this vibratol, and tissues are sliced into these thin slices in the ice cold buffer. And this will be cultured in the lab. And in addition to preparing from the tumor tissues, I was able to prepare the tissue slice culture from the cancer metastatic organs like brains or lung or liver slices, and confirm that they maintain the original tissue architecture like this. And so by utilizing this tissue slice culture system, I am observing the cancer EV transfer directly within this intact tissue architecture by introducing these highly sensitive EV levels. And now I will address these tissue-oriented questions like which are the target cell types for the cancer EVs and the spatial distributions, how far that the EVs secreted from a single cancer cell can reach, or also the tissue factors that affect the EV transfer. I first supplemented the EVs isolated from this breast cancer cell line, staying with the PK67 onto these tissue slices from the breast tumor slices and lung slices and liver slices. And I was able to observe the EV uptake by these live cells on this tissue slice culture. And the distribution, you can see that the distribution of the EVs are very uneven throughout the tissue slices. And this suggested that the EV uptake is very specific. Some of the specific cells are uptaking the cancer-derived EVs. And then I established the ex vivo model, metastatic models, by seeding the breast cancer cell lines firstly onto these tissue slices from the metastatic organs. In this case, it's brain. And these cancer cells are introduced with the Halotac fused CD63 for labeling EVs, and also introduced with the nucleus GFP. So, and this is a time-lapse image. I'm trying to. I cannot send the video actually. It's very unfortunate to have this. I don't have this. But like here, I was trying to show that these breast cancer cells on this brain tissue slices are secreting the EVs to the environment like this. And you can see all the particles surrounded the cells. And yeah, this is a two-photon microscopy image, time-lapse image of the two-photon microscopy, and I was able to observe the live, like EV secretion in live on the tissue slice culture. And further, the same cancer cells with Halo CD63 were seeded on these lung tissue slices to prepare the lung metastatic model, and it was observed at the endpoint with the confocal microscopy. And here the EVs are level, Halo CD63 were level with yellow. And these are all cancer cells because they have the nucleus, GFP. And you can see that some of the distribution of the EVs on the tissue slices. And also you can see that these cells on the lung tissue from the lung tissue slices are like preferentially incorporating cancer-derived EVs. So currently I'm trying to identify the cell types that preferentially incorporate the cancer EVs, so which are these cell types. And to summarize, I will utilize a tissue slice culture platform which allow me to directly observe the cancer EV transfer within the intact tissue architecture in real time. And I'm trying to determine the target cell preferences and the spatial distributions. And by combining with the cutting-edge analysis tools like the spatial expression analysis, so this platform will become a powerful tool to understand the biological roles of the spatial temporal deliveries of the EVs to establish the microenvironment and the metastatic niche. So lastly, I'd like to acknowledge my mentors and collaborators and the fundings to support my research. And I'd like to also add that I'm having the poster presentation on tissue slice culture work Friday, tomorrow, at poster number 503. And I'd appreciate if you stop by and discuss more about my work. And thank you very much for your attention, and I'm happy to take any questions.
Thank you, Yoshida. I have one short question. So you said that the glycosylation pattern with leptins was also impacting on going via blood barrier or going into the lungs. Have you observed any blood group dependent changes? Have you ever investigated whether certain ABO blood groups are impacting on cancer metastasis because the blood group ABO system is a like population lectins which are on the cells, in the epithelial and on the blood cells. So have you ever seen this, or have you focused on this?
Oh, thank you very much for your question. It's really interesting point. Unfortunately, I don't have any experiences on investigating the ABO, red blood cells, surface glycosylations, but the surface is very popular glycosylation patterns, and it can be really it is possible that affect the cancer metastasis or immune cell reactions as well, which is important for cancer.
Thank you. So we have a question from the audience.
Hello, Chris Reimann from La Trobe University. Thank you very much for this talk. It's a very promising role for glycoproteins. I was wondering, do you think this probably cell-dependent signature of glycoproteins could be used as sort of an address code for targeting cells in a targeted and directional manner? And what do you think of the uptake mechanism? Does the glycoprotein composition on the EV affect endocytosis, fusion, or docking?
Yeah, thank you very much. So I think it'll be really likely to the glycosylation is working on the specific structures that glycosylation is working on the cellular uptake or internalizations, and there are currently there are not so many reports on the specific structure, but there's several reports that for example heparan sulfates are involved in the cellular uptake, that it stimulates it works as a receptor to stimulate the uptake, and whereas also the integrins are on the EVs, and they are also highly glycosylated, and some groups show that the surface integrins have altered the bisecting the GalNAc, I think affect the cellular uptake of the EVs, and that affect the cancer cell metastasis. Those are a few examples, and so I think that it's promising, but still we don't know about that in detail, how the glycosylation are working on the cellular updates, and I'm really interested in that their work investing in that.
Follow-up question. Do you think in the future, if we cracked that code, that we could say a certain EV signature is targeted directly to certain organs or cells in a tropism manner?
Yeah, I think it's possible, but I also think that it's not defined by the only glycosylation. Of course, it's the merged effect of the proteins or glycosylation and lipids as well. So but I think that glycosylation will be also contributing to the biodistributions and coding, as you said.
Thank you.
Thank you. Thank you. Now it's my pleasure to hand over to what I call is a really key opinion leader in the field of EV therapeutics development. So thank you, Sakyan, for taking over the session now and for planning this together. It's always a funny and very exciting way of interaction we have. Thank you.
Thank you, Eva. So I think most of you appreciate the theme of where we are going. Basically, what we want to know is what makes the EV goes to where it goes, where you inject, how you modulate the EV. So I think the next speaker is going to continue with this theme. So we have Luis Lima. She'll be talking on the cytokine binding to SEBs directs metastatic dissemination in breast cancer. Dr. Lima is a research officer in the QIMR in Berhover. So, please.
So, thank you very much, Dr. Sarikian, for the opportunity to discuss my work here with you guys during this session. So today I will be presenting some of the data that I obtained during my postdoc in the Tumor Microenvironment Lab in QIMR. So these data altogether, it indicates that the binding of different cytokines to small extracellular vesicles can determine like the biodistribution of CAVs and ultimately like direct the metastatic dissemination in breast cancer. So I think we all can agree that in most types of cancer, the patient mortality is largely caused by the metastatic spread of tumor cells from like the primary tumor to distant secondary organs. And this is especially because of the lack of efficient treatments that target specifically like the biology of metastatic tumors. So it's really important for us to understand this whole process so we can improve the clinical management of metastatic patients. Whether it's like extracellular vesicles, how they are involved in this process. So we know that EVs, they are involved in like in different aspects of metastasis, and one of these aspects, aspects is the formation of what we call the premetastatic niches. So we know that the primary tumor, including like cancer cells and other non-cancer cells such as immune cells and other stromal cells, they can release like cytokines, growth factors, and other soluble factors. And together with the EVs, all these factors, they can prime distant secondary organs and creating a favorable niche that will facilitate the development of metastases in these secondary organs. We also know that the formation of this premetastatic niche mediated by the EVs, it can occur in a like organ-specific manner. So it has been shown that the integrin profile of small extracellular vesicles can determine the accumulation of these EVs in different organs. For example, like EVs that are positive for alpha-6, beta-1, 4 integrins, they will preferentially like accumulate in the lungs, while like alpha-V, beta-5 SEVs, they will accumulate in the liver, creating premetastatic niches in these organs and ultimately like leading to the development of organ-specific metastases. We actually wondered if not only like intrinsic molecules of EV such as the integrins, but if molecules presenting the tumor microenvironment as soluble factors, they could also participate in this EV-mediated cancer organotropism. So would it be possible that all these factors, they would like be associated to then determine like the formation of these premetastatic niches and consequently like influence metastasis? So to answer that question, we used like breast cancer EVs. We isolated these breast cancer EVs from conditioned media of cell cultures or from plasma samples of like breast cancer patients. For that, we used a combination of ultracentrifugation and size exclusion chromatography in the case of cell culture derived EVs, or size exclusion chromatography directly in the case of the plasma samples. We characterized these breast cancer EVs by transmission electron microscopy, so we can see here the morphology of these EVs isolated from mouse and human breast cancer cell lines, and also from the plasma of healthy subjects and breast cancer patients. We determined like the size distribution of these vesicles by TRBS. and we can observe that the size range is corresponding to what we would call like small EVs, but I will just call them EVs during this presentation. We also confirmed the expression of EV-associated like protein markers by Western blots, such as HSP70, TS011, CD9, and Flotalin. So after characterizing these isolated EVs, our very first result that actually started like this project was we observed that EVs that were isolated from the blood of breast cancer patients, they were co-purified with a large range of cytokines. We could actually find cytokines in EVs purified from the plasma of age-matched healthy subjects as well, but this was much more pronounced in the patient samples. As you can see here in this heat map that we use just to quantify these cytokine profile arrays, CCL2 was one of the cytokines that were found to be like in high levels in the patient samples compared to the healthy subjects, and we confirmed the high levels of these cytokines in the EVs of patient samples by ELISA. In contrast, when we isolated EVs from cell culture of like breast cancer cell lines, we observed they were devoid of cytokines. So we then like asked ourselves if these cancer-derived EVs, specifically if they could be labeled by cytokines present in the tumor microenvironment, and then explain the large range of cytokines we found in the blood of cancer patient samples. So what we did was we induced primary tumors, also topic like breast cancer tumors into syngeneic mice. After these tumors grew, we harvested these tumors, put them into culture. We then collected the conditional video of these minced tumor tissues. We processed it to deplete them of EVs, and then we assessed the cytokine profile of what we call this tumor interstitial fluid. And as you can see here in the cytokine profile array, and also quantified by ELISA, there is like a range, a number of different cytokines present in this tumor interstitial fluid, including again like CCL2. So what we did next, we incubated the cell culture derived breast cancer EVs, which again were devoid of cytokines with this tumor interstitial fluid. So we incubated there, we re-isolated these EVs by size exclusion columns, and then we assessed whatever cytokine would be bound to these EVs. And as you can see here by ELISA quantification on the black bars of the EVs incubated with TIF, these EVs could be bound like by different cytokines, but again like especially CCL2. So we did some functional work using TIF, but as a proof of concept, we used CCL2 to simplify our model, and I will show here today like our results with CCL2. So we incubated like recombinant protein like mouse or human CCL2 with EVs isolated from mouse and human breast cancer cell lines, and we can see that the binding of CCL2 occurs in a dose-dependent manner, although at different levels depending on the source of the EVs. One of the things we really wanted to know is the localization of the cytokine, because if it was located on the surface of the EVs, it would definitely impact the function and the targeting of these EVs. So as you can see here, after incubating the EVs with CCL2 and submitting them to assessment by ELISA, we observed no difference between intact EVs, or if we ELISA the EVs before the ELISA experiment, suggesting that at least most of the CCL2 would be on the surface of the EVs. We also treated these EVs after the binding of CCL2, we treated them with proteinase K in a concentration that would remove any surface proteins bound to these EVs. And as you can see, the proteinase K treatment reduced the amount of CCL2 bound to the EVs, also suggesting the localization, the surface localization of these cytokines. finally, we really wanted to corroborate this, so we used surface-enhanced Raman spectroscopy to compare EVs conjugated or not with CCL2. And this technique is able to determine molecular fingerprints of membrane proteins on the EVs. And as you can see here, we were able to find different Raman scattering spectrums in these two samples, and we were able to clearly distinguish them by principal component analysis, suggesting that CCL2 was indeed on the surface of these EVs. So after that, we asked ourselves, how do the cytokines, or specifically CCL2, bind into these EVs? Interestingly, when we did the proteomic assessment of these breast cancer-derived EVs, we could not find most of the cognate cytokine receptors, including CCR2, would be what's the receptor for CCL2. However, we could find a large number of different proteoglycans, which were differentially expressed in the breast cancer-derived EVs, depending on the aggressiveness and metastatic potential of the breast cancer cell line. And so, because it has been shown in the literature that proteoglycans, they can bind chemokines and cytokines in different biological contexts, we investigated if this could happen in the EV context as well. And indeed, we were able to co-immune precipitate different proteoglycans such as versicans, INDECAN-1, CD44, HSPG2 with EV-bound CCL2. We also treated the EVs with enzymes that are able to degrade glycosaminoglycans chains of heparan sulfate and chondroitin sulfate present in these proteoglycans. And we can observe that this treatment reduced the binding of CCL2 to these EVs, indicating participation of the proteoglycans on this mechanism, on this process. Finally, we wanted to see how the cytokine labeling of the tumor-derived EVs could affect their biodistribution in vivo. So what we did, we intravenously inject syngeneic mouse with fluorescently labeled EVs, and after 24 hours we harvested some tissues and organs such as liver, spleen, kidney, lung, heart, and bone marrow of these mice. And as we can see here, the EVs that were conjugated with CCL2, they were preferentially accumulated in the lung of these mice, and this is confirmed by fluorescent microscopy of lung tissue sections, as you can see here. Also, we processed these lung tissues to obtain single-cell suspensions, and we looked at specifically lung immune cells, because we observed that lung leukocytes, they are frequently positive for CCR2, the cytokine receptor. So we, as you can see here, the CCL2 conjugated EVs, they are more pronouncedly taken up by CCR2 positive cells, presenting this total immune cell population, and this difference could not be observed in the CCR2 negative population, suggesting the participation of this receptor in the targeting of these EVs into the lung. Interestingly, when we used CCR2 knockout mice, we could not see a difference in the retention of these CCL2 conjugated EVs in the lung, nor we couldn't observe any difference in the uptake of these CCL2 conjugated EVs by the immune cells present in the lung of these CCR2 knockout mice. Again, really strongly indicating that this receptor was involved in the retention of these EVs. Finally, we wanted to know if these CCL2 conjugated breast cancer EVs, they could favor metastasis of breast cancer cells. So what we did, we conditioned mice, syngeneic mice, by repeated injections of conjugated CCL2 conjugated or unconjugated EVs, followed by the injection of intravenous injection of the breast cancer cells themselves. And after 2 weeks, we collected the lungs from these mice. And as you can see here in wild-type mice, the CCL2 conjugated EVs, they significantly increase the metastatic establishment of these breast cancer cells in the lungs, which was not observed in the CCR2 knockout mice. So in summary, so this is part of the data we obtained during this project. We did some experiments with the more complex tumor interstitial fluid conjugated EVs as well. I think we were able to demonstrate that soluble factors, or specifically cytokines that are present in the tumor microenvironment, they can decorate cancer-derived EVs, mostly via the binding to proteoglycans present on the surface of these EVs and the glycosaminoglycan side chains present in these molecules. So this cytokine decoration of EVs can influence the targeting of these EVs into different organs. In this case, we observed an increased EV retention in the lungs, and these EVs will specifically target to cells that present the CCL2, the cytokine receptor, CCR2. I didn't show to you guys, but we observed that when you looked into further details into these immune cells, NK cells and myeloid-derived suppressor cells were the main targets of these EVs. And finally, we believe that the interaction of these EVs with these cells can create niche that favors metastatic outgrowth and ultimately influence or increase the chance of metastasis by this mechanism. So I would like to thank my whole group, which is actually mostly relocated to Hong Kong now, together with our PI, Andreas Miller. So I'm still in QIMR, but I'm moving there soon as well. I would like to thank the former members of the Tumor Microenvironment Lab who participated in this study, all our collaborators, the funding, and especially thank you for your attention, for resisting through this long day. Thank you so much.
So, thank you. So, as this is a session strongly focusing on therapeutic approaches, so what was now your concept thinking of therapies? How would you go and interfere with this CCL2, CCR2 axis, or what is your therapeutic idea behind the nice data you showed?
Actually, the initial idea about this project was actually trying to determine an address code, as we discussed here before. So we imagined that if we could find a signature of cytokines bound to the EVs in cancer patient samples, we could try to predict which would be the organ affected by metastatic cells in that case. So actually we found out it's not that easy because we would need a high number of patient samples to try to reach statistically significant profile signature to determine. And we also, as we discussed here, it's not just the cytokine profile, other molecules are certainly involved in this targeting. And another thing that we're actually interested in trying to check if the proteoglycan profile of the EVs can also predict the organ that the tumor cells are going to metastasize. So
only a short comment, because many EV research groups have shown accumulation in lung and liver, so not really a surprise, huh?
yeah. Next question.
Yes, very interesting. I was wondering, because I understood the primary role of cytokines is to attract immune cells. Is that still retained? Is that activity still present when it's bound and captured by EVs? So does it still attract immune cells and maybe divert it from the primary zone of inflammation?
Yeah, actually, I didn't show here, but when we intubated the breast cancer-derived EVs with the tumor interstitial fluid, and then we injected into mice, and after the conditioning, before injecting the tumor cells, we checked the immune composition of the lungs, and we could observe like a change in the frequency of different immune cell subsets. So that could possibly like collaborate to the creation of this pre-metastatic law, immunosuppressive like pre-metastatic niche.
Thanks.
I think one more question.
Just a general philosophical question for the room. Is there any attempt to understand like the music theory behind all of this? Because we can presume that when you get cancer, there's clinical response, so that there would be some just incidental bystander cytokines. There would be some that are actually trying to help put away the cancer. I think inherent in all our searches is that we think that exosomes are, there's something pathognomonic or specific or identifiable through machine learning, but it's kind of like data mining too. We can't really assume that everyone at the scene of an arson is responsible for the fire, right? So I think that's the kind of worrisome thought I have when I hear these talks. Like, we know the answer's there. We know the cancer exosomes are all bad, but are they really? Maybe they're mostly good, and maybe most of these cytokines are mostly good. So that's, it made me think of Dr. Lee's, you know, there's a life cycle to cancer. You know, there are different responses to the immune system. So I just worry that we're oversimplifying, that we're always looking for that needle in the haystack. Maybe there's a lot of needles and there's a lot of different types of hay.
Yes. Actually, like I can just comment, not for this project, but we have some data in the lab where, depending on the source of the, specifically like tumor-derived EVs, depending on the protocol of conditioning that we use, the amount of EVs, we can actually use EVs to reach like a anti-tumoral effect. So we have some models that we see the opposite. Like actually, if we condition the mice with repeated injections of EVs, and then we injected the tumor cells, we have reduction of metastasis. So I think it really depends. Like,
Thank you very much for interesting talk. To answer the last, the last question from the member of the audience. So you have heard about all these different things that where you inject the EVs, what you do to the EVs can change the distribution. But I think ultimately we need to know the baseline distribution first. And I think our next speaker, Anthony Wu, will do just that. Okay? Just have to get his title out. So his title will be Beyond the Effectiveness, Where and How do Extracellular Vesicles Distribute in the Living Organ. Anthony Wu from the Academic Clinical Taiwan.
Thank you for your kind introduction, and thank you for giving me this chance to be presented here. And thank you everyone to stay till this long day. And from my title, it's maybe it's not easy to get into everything from my title, but hopefully after this talk, I hope I can convince you that instead of local biodistribution, the systemical biodistribution should be also considered as one of the key parameters to be studied. So let's start with a very basic, for today or yesterday education day. We already have a lot of good introductory for the different subpopulations. So we have exosomes and exosomes and different subpopulations such as apoptotic bodies. And then lastly, there's a lot of different other vesicles has been identified. And then based on the categorization, we can generally, based on isolation method or the size, we can generally categorize into maybe small EVs and large EVs. And then these EVs plays a different roles in for the applications. For this one, EVs carries a lot of DNA, RNAs, and proteins, lipids such as glycans, and then responsible for a lot of different functions. And then here we have use it for the diagnosis, and as well as for therapy, and then for drug delivery system. I think I will put more emphasis on these two. And then for the delivery system or therapy, when we inject it exogenously, and then how is it beyond the effectiveness? So equal to my title is like effectiveness is very important, but beyond the effectiveness, how it distribute in our body and how it systemically affect the whole organ distribution is quite important. So throughout the topic, I will generally guide through the like EV targeting, which like we change like surface protein to alter is like organotropism, and then like EV dosage, how EV dosage will affect the biodistribution, or EV size. And lastly, different routes of EV administration may also affect the overall distribution. So there's like different level limits has been proposed, like such as chemical synthesis type, and then pKh, DIL, and derivative, or CSFE. Those lipophilic type, or the other like CSFE. And the other is radioactive tracers, and then lastly will be genetic modified reporters. And then a lot of different reporters has been proposed and then been used. And then like what Charles introduced during the education day, and then based on there's no level of these like reporters or labeling method. It's just like based on what kind of situation or what kind of like modality you want to use. So, but the general rule of thumb is like when you are thinking of this labeling method, spatial temporal accuracy or signal to noise ratio of this reporter or accessibility, such as maybe radioactive tracer, is very good for the in vivo study. However, maybe it's not easy to access, or such as for maybe some lab, it's not producing maybe lentivirus or something, then genetic modified reporters for stable line, that would be a concern. So after that, so today's talk I will focusing on more on my work is like PalmBread, which is a bioluminescence resonance energy transfer based EV reporter that I have developed back to like 2020. I have published in 2020. And then basically this reporter is comprised of like commutation signal, and then to the N-terminus of the GFP fused with the nanoluciferase. And then this system is similar to FRET. And then once it catalyzes its substrate or NLUC, here we use like fluorofluorine or fluorizine. And then it will generate different modality of the intensity. First one will be the bioluminescence intensity, which will generate the bioluminescence intensity around like blue light range. And then it will resonate, its internal light source will serve as a resonance energy transfer to the fused GFP, and then induce the bioluminescence so-called bread-induced fluorescence signal. And then lastly, don't forget that we can also use like external light source such as a microscope to excite the GFP to have the third modality of the imaging. And then once it's like, after its post-transcriptional modification, it will be modified in palmitic acid, and then this palmitic acid will insert into the inner leaflet. And then we have demonstrated that this reporter has predominantly labeled inner membrane of the EV. So basically, what we have done to achieve is like we use this reporter to achieve like multi-resolution PAN-EP imaging, as well as this PAN-EV or EV particle labeling. Since we also use AF4 that has also can isolate by different population from small EVs, and then we can also detect a signal from the exometer fraction. And after that, so let's stop. Sorry. And then with this data, I want to like introduce like how is like multimodal or multi-resolution. So here we use like inject the 2950 PalmBread EV through tail vein, and then imaging with a different time point. And so after we inject our EV, and then after around 4 minutes, we will inject the substrate, and then to imaging lively. So as you can see here, we imaging from 5 minutes all the way to 30 minutes. And then within 5-10 minutes, the signal almost gone for the bioluminescence signal. And then this one we gate with the GFP filter to have the signal. So we can see that it shows the less signal, but it shows more clean signal from like we can see more in the arm. But however, when we use like excited fluorescence to try to excite the RGLP, it shows a very high signal background here. And we can also see the same modality from the ex vivo. So after 30 minutes, we took out all the organs and immersed into the substrate. And then with this immersion, we can also imagine for the bioluminescence and then brain-induced fluorescence, as well as excited fluorescence. So again, compared to a brain-induced fluorescence, the excited fluorescence seems to give us like more false signal. And then all the way after this one, we can see that from the ex vivo part, we can also do the IHC as an orthogonal assay. So here I show the lung as one of the representative. So EP, green is for EP, and then blue stands for the nucleus, and then merge with the DIC. And then we can use like, we can compare to the PBS, and then see here the quantity. We can do the semi-quantification by using the antibody tagging, and then using the super resolution method to quantify these extracellular particles. So we can see that corresponding to the ex vivo data, we can see that there's a lung followed by spleen and liver and kidney distribution by injecting the 293T EV, pump-bred EVs. So this one, after this one, I just want to use this one to convince that the pump-bred enables the multimodal and multi-resolution EP imaging and semi-quantification. So let's start with like for today's. I want to introduce different kind of modality and then different importance of the start from the EV organotropism. So here we try to modify the EV. Since in this project we use like not only use the 293T, we also study for the mouse liver cancer. So with this mouse liver cancer, we use like knockdown assay to knock down the surface protein from these liver cancer. And then after that, we use like scramble as a wild type. And then followed by this one, we use the education assay. After 74, we compare like 0.5 hour post-injection, and then all the way inject education to the 72.5 hours. So as you can see here, we collect all different major organs, including brain, lung, heart, liver, and then spleen. So as you can see, compared to the scramble, mostly since this mouse liver cancer has been, from other data, we've already demonstrated that it has a long tropism, and it goes along more. So as you can see here for the distribution, it shows more in the long accumulation in the tropism. And then after knockdown of SLC21, one of the ligand identified by proteomics of the HCA1 derived EPs. And then it is suggested to that it will related to the lung metastasis. So we knock down the SLC21, so we can see that it do reduce the tropism of the HCA1 derived EP to the lung. And then not only that, we can also see that there's an overall change in kidney. It increased the kidney accumulation, as well as here is like it also have a liver increasement after 72.5 hour of the education. So first thing is that when we modify the surface ligand, you will also change not only the targeted sites, such as lung here, but also you will also change the overall, which is the systemic biodistribution of the E. And then later we also use this one from my colleague, and then we use also use the pump-bred as a system to express in a triple negative cancer cell for the 14-1. And then we use the biopsy as a syngeneic model to inject the 41 pump-bred. And here we use like the double labeling also with the DIR. And then here we test with different dosage and then as well as the size. Here the BEV, not to confuse with the bacterial EV. Here the BEV is a collective term of the medium and large EV, which we set it has a mean size generally bigger than 200 nanometer. And SEV, small EV here, we set it as smaller than 200 nanometer. So here we also use, with a bolus injection, after 0.5 hour, we have a one-time point, and then sacrifice at 24.5 hour as well. So here we can see that from the different dosage, here we show that 10 and 25 micrograms. See, we can go into the single population. So as you can see here, generally it's like there's a dose-dependent increase of like when you inject more of the EV, you can see that it has more accumulation to the lung here. And then so we can find in the SEEV at 0.5 hour. So here we introduce like there's a dosage-dependent EV biodistribution differences. And not only that, overall distribution changes is also we can find from the liver. We can see that there's a liver differences within the population, such as like after 24.5 hours, it goes to the liver more compared to the lung. So here again, like our reporter shows a very good sensitivity in reporting these systemic biodistribution. And then here we also introduce the DIR as a dual level. Since here we want to compare that if the DIR can report this biodistribution. But unfortunately, it's like here we can see that mostly we can only report mostly goes to the liver, and then cannot actually report the other biodistribution from the other organ. And then the other things is that we found throughout this experiment, we found that the EV dosage, if you increase like the reason why we only show the 10 and 25 microgram here is like when we inject higher than the 50 or even to 50 or 100 nanogram, EV do kill the mice after we injected it. After we injected it, within like 30 minutes, it shows a very strong like choking symptom. And then within like a half day, they just die by EV injection. So, but maybe this is very cell type specific, but we also found this kind of effect. So EV dosage do matters. And then later, since we also compare different sizes, it's like we compare the BEV and SEV. So here we use like the 41 pound grade, and then here we also inject the 41 cells as a model to test. And here we use a different education method. So first, at day 0, we inject the 41 cells, and then to establish the syngeneic model. And then at day 0, we also inject different BEV and SEV. Here we also do a different surface modification. Try to see that the different, if the surface modification of this BEV or SEV will affect the tumorigenesis. And then here, for the tumorigenesis, here I will more focusing on the biodistribution. So as you can see here, we sacrifice the mice at day 0 as a background, and day 14 as a mid phase, and then 18 days as a late phase. And we collect the tumor and then blood, including different organs, and then do the biodistribution analysis. As you can see here, the BEV and SEV shows the in the initial and then the mid-phase differences, including like we can see that there's a liver distribution difference initially compared BEV to the SEV. And then for the CD44 knockdown here, we see as CD44 here is also identified by proteomic analysis. It's one of the potential ligand that leads to the long tropism. So here we can see that by knocking down of the CD44, we also have the reduce. First is like reduce of the long tropism. Not only that, the blood retention is reduced compared to BEV and SEV. SEV has a reduced lung retention compared to the BEV. And then lastly is like from chemical group, they tested for the different pharmacokinetic by injecting the EV expressing our reporter. They tested in the non-human primate as a very landmark paper. So they tested non-human primate and then collect the different organ with a different injection routes. So as you can see here, it's like compared to the IV injection one, there's a different biodistribution throughout different organs, they collected including liver, spleen, kidney, and lung, and that as well as the heart and colon. And then they further dissect into the brain to see like if the different route will affect the biodistribution. And then the answer is yes, and then they also found that the more is like goes to the medulla. And all in all, I would want to convince you or convey an idea is like for the drug delivery system development or like therapy, EV as a therapy cargo, beyond effectiveness, we can find that a lot of work has done in the EV effective and it work. And however, however, the systemic biodistribution change, which means that instead of the targeting site or like the disease we are going to cure, how about maybe the other changing distribution may generate some side effect potentially. Then so here we go into different aspects, including the EV targeting, organotropism, and the EV dosage, EV sizes, and the route of EV administration should take into consideration. And after that, hopefully for more like pharmacokinetic analysis, as well as where these EV goes and can lead to the more detailed mechanism of action studies. And then lastly, there's a different review, and then position paper, and as well as a review suggests that a different modality in EV therapy and the potential of the EV therapy. And then lastly is like I have recently developed a reporter that can simultaneously tracking of the different populations. Say here is small EV and BEV population, and then I will have an all-round introduce on Saturday. And then lastly, I want to thanks to my lab and then my PI Charles, and then all the members in the lab. And then thank you for your attention, and then I would like to take any question.
Nice talk. I have a question about the mortality that you mentioned with high dose. Can you elaborate? Was it IV or IP?
It's IV injection.
IV injection, and then how soon the animal died?
Since I remember that time is back to the pandemic, and then we just communicate with to our collaborators through line, and then we found they reported to us that soon after injection, within 5 to 10 minutes, they found this symptom.
Symptom, okay.
Well, it's just like choking. It seems like it's choking. The breathing is like
The breathing difficulty.
Yeah.
So do you have any hypothesis of what happened?
Maybe just like we have since previously we have reported. So we previously when we found this one, since back to the HCA1, the one liver from mouse liver cancer, we did not see this kind of effect. 250 we also found in the 100 microgram half. And then after that we soon dissected and then used like ex vivo assay. We found that most of the signal was accumulated in the lung. Actually, ED signal was accumulated in the lung. And then I think it's because like viscosity of for the 41 maybe it's due to its viscosity. Yeah, it's quite viscous. And then for the ED, so I think
perhaps a large amount of ED could have blocked the airway.
Maybe capillary
alveolar level.
Yes, maybe like maybe not only airway like capillary of the lung, and it just reduces oxygen exchange.
Do you think any EV could do that?
We only observe it maybe 290 and then the 41, so it might be cell type specific, and then because as what I said, maybe it's due to the viscosity. So maybe it's not all the EV cell type direct EV will have this kind of issue. So, but for the dosage, I think that's one of the key issues when we are developing some therapeutic strategy.
Yeah, thank you.
Thank you for your last presentation. Actually, for Professor's question in my lab, in our lab, we also made some problem after we injected the cerebral EV contain CRISPR-Cas9, our mice died, and we go back to do some. We removed the endotoxicity of the plasma. Then we injected again. Then the mice survived. So we think maybe there is some endotoxin problem happening in the cerebral EVs. Okay, I'll go with my question. I have one question about the construction of your pump grade. For those pump methylation peptide, can I know those peptides derived from which protein?
It's neuromodulating, GAP43, and it is first 20 amino acid.
Okay.
Yes, human neuromodulating, GAP43.
How you select this peptide at the beginning?
I mean, this one is based on the previously published in Nature Communication POM GFP and then POM detonator is back to Charles' work. And I believe that he selected from discussion, and then he selected from by discussion with their lab mate, and then after that he found that it can label the inner membrane, and then as well as combinatorial signal is quite common in signaling transduction pathway. So I think it's quite good for the labeling on the membrane.
Okay, so you think that those GFP is inside of the EV or on the membrane of the EV?
We have in our articles there's a lot of detail that I did not show. We use dot plot analysis with and without detergent to demonstrate that our reporter are predominantly labeled in their membrane of it.
Okay, thank you. Thank you, Lester.
No more questions? I hope you have a good understanding of the talks in this session. I hope the take-home message is that where you inject your EV, what you do to your EV has an impact on where it goes eventually. So when designing experiment, please keep this in mind. I hope you all got the message, and thank you very much.